Hello, everyone, and welcome to today's webinar, Linking Spatial and Temporal Profiling to Clinical Settings Through Advanced Machine Learning. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Vizgen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers, Dr. Carole Chedid, Field Application Scientist, Vizgen, EMEA, and Henrik Heiland, Ph.D., Full Professor of Translational Neurosurgery at Friedrich-Alexander University, Erlangen, and Attending Neurosurgeon at the University Hospital Erlangen. For a complete biography on our speakers, please click on the Presenter tab. Carole, Henrik, thank you for joining us. You may now begin your presentation.
Hello everyone, thanks for joining us for today's webinar. My name is Carole, I'm a Field Application Scientist at Vizgen. I'm based in the UK. And today I'm just going to give you a brief introduction on who we are and what we do before leaving the floor to Henrik for his talk. That's to give you a little bit of context. Our company is headquartered in Cambridge, Massachusetts in the United States. And it was founded in 2019 as a spin-out of Harvard University from the lab of Xiaowei Zhuang. And as of today, we have over 200 employees worldwide. And our mission is truly to pioneer the next generation of spatial genomics, really go into uncharted territory here and provide researchers with tools that will set a new standard, kind of push all the possible boundaries and really go demonstrate the possibilities of what can be achieved with in situ single cell spatial genomics. So through these, we're enabling researchers to gain new insights into the biological systems that govern both human health and disease with spatial context. So why spatial genomics? Well, the importance of spatial organization, information, and context is quite easy to appreciate if you look at it from a macro level. For example, if you look at these tiles separately, it doesn't make much sense just looking at them like this. Whereas while you look at them with their spatial context, it's quite different from reading them in their original sequence or pattern that was picked up. So really, information without the spatial context doesn't truly offer the insight it could. And the same is true for biological system. We can understand that a defined structure is necessary in order for the organism to function. So if we move from this metaphor to something a bit more applied and we look at things a bit historically, we can look at advances in RNA sequencing, and starting off with bulk RNA sequencing, which was a huge step forward and helped us understand the expression of many genes at once in biological system, but let us truly appreciate differences between tissues or between different states. And from there, having the extra layer of information of single cell really let us break this down to a whole new level of granularity and detail and understand why some particular cells are contributing to some gene expression levels within the tissue. So from there, we can understand what extra, level of depth and information is brought by single cell spatial genomics. And this is where Vizgen come into place by offering highly multiplex direct in situ detection. So to give you a little bit more insight into what the spatial genomics data that you would get off your microscope would look like, let's view it as really a transcriptomic and subcellular tissue map. You would have really different scales, right? So let's say you've got this map of New York, If you look at this map as is, you would kind of understand the general area, the vicinity, the location, but not really any specifics of the area. So we could compare this to a whole tissue view. You can see your structure, you kind of know where you are, you can see the main factors, but you don't have any details yet. So if you zoom in a bit more, you can start to see that, okay, you're in Manhattan, you can see the park. This is what would be compared to a wide field of view kind of overview of the tissue, where you can start seeing cells, interactions, and cell functions. And you can actually continue to zoom down until you get to a small 1 by 1 meter area on the corner of 2 different streets, find out whatever this is, Apple Store, pizza restaurant, I don't know. But this would be what we would see really at the subcellular level on our data. So what does this actually look like now? Animation is just showing data from one of our most well-described tissues, which is the mouse brain, and really lets you see the connections from whole tissue to wide field of view, which is about 200 by 200 micron, and then all the way down to subcellular, where you can start seeing neuron shapes, you can start seeing specific transcript just by eye, where they're restricted within the cells, different cells altogether. So just to go into a little bit more detail into what the chemistry behind all of this is, I just want to clarify some things here. We work with a chemistry called MERFISH, and MERScope itself is simply the easy-to-use, fully automated platform that Vizgen has put together to make MERFISH chemistry accessible to users. So there's loads and loads of MERFISH papers already existing, and what we do is kind of package all of that in for ease of use. So just as a quick reminder, MERFISH combines the accuracy of direct in situ RNA quantification from single molecule fish with a barcoding system which lets people create custom gene panels of currently 1,000 plex. And you get this with high accuracy, high consistency due to an error robustness correction system which is embedded in the decoding itself. So this is why it stands for multiplexed error robust fluorescent in situ hybridization. What we've tried to do is really provide users with a fully comprehensive end-to-end platform solution that starts with gene panel design that can be fully customized and can be done via our online portal, to sample preparation, where we're here to provide support, and every type of region is pre-packed into convenient and easy to use kits, all the way down to actual data acquisition and the instrument that you can see here, all downstream to data visualization and analysis that can be performed via our completely free visualization software that lets you kind of see your fluorescence images, your transcripts, your UMaps, your cell segmentation mask, and is all in all a very comprehensive tool to really take you all the way from the bench to your data. Important point to note, which comes straight from the nature of MERFISH chemistry, is the flexibility that our system has. We've proven MERFISH work with our framework in over 70 tissue types from mouse and humans, but we're also able to analyze tissues from any organism. We've got work being done on plants, on zebrafish, on xenopus, Drosophila, all sorts of non-model organisms are being analyzed. And sample types that are compatible with the system include cells, fresh or fixed frozen tissue, as well as FFPE tissue blocks. And this, of course, is being as well validated in the field. As of the end of last year, 2023, we passed the bar of over 200 peer-reviewed publication and preprints in a variety of journals, and the institutions that are using MERFISH and MERScope keep getting more and more numerous and testing out new protocols, new processes. So just quickly, without getting into any sort of technical details, I just want to finish on a few takeaway points that explain how MERScope was designed from the ground up for data accuracy. And this starts at the sample prep level. One core aspect of our sample prep processes is tissue clearing. So that means that we are going to embed the tissue into a gel, and then this is going to lock the nucleic acids in, and we're going to clear away all of the protein and lipids, which would create autofluorescence in backgrounds. And this really improves the signal to noise ratio, and this in turn helps decode the transcript Murphy spots. And this is being used, of course, in conjunction with hardware. So the microscopy aspect of the instrument has been worked on really hard, and we offer superior optical resolution by using a 60x objective lens and a high numerical aperture, which means that basically every transcript appears kind of smaller. So using this together with an algorithm, a localization algorithm at the end, means that the resolution on the digital image at the end is 100 nanometers. In terms of chemistry, we rely on a multi-site, multiple probe chemistry for target detection, and this includes redundancy and overlap. So that means that the probes are going to tile across the transcript, which means that it's going to increase specificity, and we won't need any sort of amplification to see the signal at the end. And finally, on the bioinformatics aspect, the error robustness that I talked about at the beginning of this little presentation is going to help ensure that even if there's an error while decoding, a mishybridization event, something goes wrong, the system is still able to correct that and find what was the intended gene barcode created in here, and helps really increase the accuracy even with a high multiplexing. So each of these separate design features play an important role in increasing both the sensitivity and the specificity of the data collected. Well, that's it for me. I will leave the floor for Henrik to give us his presentation, and if you need anything, have any questions, we're happy to discuss in a Q&A session at the end, and of course, don't hesitate to reach out for any queries. Thank you.
First of all, I want to thank the organizer of this webinar for the opportunity to share some of our data, how we use spatial transcriptomics and spatial data in general to bridging the gap towards clinical applications, how we can use these data to really make clinical decisions in the future. So nothing to declare. The disease I want to talk today about is glioblastoma, which is the most malignant tumor of the central nervous system. It's a rare tumor, but highly malignant. So most of the patients are dying within 14 to 21 months. And so the very unique feature of this glioblastoma is that this disease is exclusively linked to the central nervous system. So even if this is a high malignant tumor, we see most of the metastasis that happens from these tumors occur within the ZNS or spinal cord. Over the last years, my lab try to explore using spatial transcriptomic data how the tumor architecture is constructed in these tumors and what are the difference and heterogeneity that we see across cohorts of patients. And we found that there are unique areas that are highly linked to brain developmental stages, such as oligodendrocytic origins, neuronal origins, or radial glial cells. But then there's also distinct areas we call areas with reactive programs that can be associated either to hypoxia changes, metabolic alterations that we can see in these areas, or they are linked to specific immunophenotypes, immune-associated programs with a higher abundance of T cells, macrophages, or activated microglia cells. The problem of this disease that we looked into mostly on the de novo stage is now to understand what happens now between this de novo stage in the temporal trajectory towards the recurrent stage of the disease. And this problem is very complex because there happens a lot of genetic changes. There is plasticity, transcriptional plasticity, also epigenetic changes that happens from the tumor perspective itself, so from the tumor biology perspective, but then there's also a lot of cells that coming into the brain and that shaping these tumors, immune cells, but also cells from the brain itself, neurons, oligodendrocytes, that really have an impact here how the tumor is growing. So we wanted to try to solve these very complex problems by using AI approaches to understand how these tumors develop over time. The problems of using AI approaches or advanced machine learning in these contexts is that we have a high-dimensional data on single-cell level, on spatial level, on molecular informations in general, and we need to link these vast amount of informations towards one clinical endpoint that could be the stage of the tumor, anatomical locations, or whether these patients received a specific therapy. And right now, there is a lack of predictive models that we can use now to really connect these data together. And there is another problem of a high variability and interpatient heterogeneity that we have to overcome. So for this, we developed a new toolbox, what we call Deep Sparta, a toolbox which has a couple of different AI approaches that we can use now to integrate multiple data sources and really understand how this works together. And one of the specific inputs I want to talk today about is the spatial data, which we want to connect to clinical informations. So to give a little bit an overview of the study design, we used glioblastoma tumors from different time points and tumor stages. And then we used two different types of data. One is Vizgen data. I want to talk first about how we use and approach these data. And then we connected this to MERSCOPE in situ sequencing data. So both of these data types are basically used to identify tumor microenvironments, and these tumor microenvironments then being predicted by AI models, whether these microenvironments derive from the different time points of the disease. And when we understand how the computer can now predict the different time points and the different stages of the tumor, we can then use and manipulate these microenvironments to really get an idea how these cellular behaviors and cellular compositions of these microenvironments have an impact on the stage and the time point of the disease. So these models are basically always working in a very similar manner. We have spatial distributed data that can be Vizgen spots or that can be cells from the in situ sequencing. And so first of all, we isolate microenvironments, neighborhoods, which containing a center cell, and then we calculate the neighborhoods, the first, second, third, and so on, order of neighborhood cells, and this we define as microenvironments. And so these microenvironments now containing the information of each node and its transcriptional identity, and also containing the information of their spatial representation, of their specific neighborhoods, or the distance, for example, of cells to each other. So we can use both of these information to train graph neural networks, and in this context here, attention-based neural networks with multiple attention heads that then reproduce or basically project these data into a latent space that we can then use to specifically predict whether these latent spaces are connected to the different time points of the tumor. One of the problems is that multiple microenvironments, for example, are being extracted and having the same endpoint and the same patient. So we wanted to optimize the model, not predicting specifically the patient identity, but rather more predicting the specific time points that we want to look into. So we also used a discriminator then to specifically being trained for the confounders that we know, and that can be patients, that can be specifically histological regions, for example, that we know. And whenever a decision was made towards the confounder, we basically penalty the loss of the real network that should predict the time points. And with this kind of network architecture, we can then precisely predict whether these microenvironments are more derived from a de novo or the recurrent stage of the tumor. But it's very complex to understand now how these networks make decisions. So now we basically trying to overcome this challenge by using explainable AI to look really into the models and understand how these decisions were made. So where are the importance were put into which cells are important, which connections of cells are important as a passive method. And then we also look into active methods, so how we can actively change the tumor microenvironment to really understand how cellular compositions have any impact on the decision that is made by the network. So first of all, we want to talk about this first approach of active changes and in silico perturbations that we can use. So here we have an example of a Vizgen sample of a primary tumor, and we trained the network, as I explained before, to now predict whether the microenvironment, so a subset of these samples, are being more likely to derive from a primary or a recurrent tumor. But as we know, these tumors are very heterogeneous, so not all areas of the tumor are directly predicted as being a primary or recurrent. So we can even see that in this specific sample here, we have areas more in green that are more likely to look like recurrent tumors, and there's other areas that are very specifically looking as primary tumors. So we can now look into the microenvironments and the cellular compositions of these areas, and here we see 3 examples of microenvironments highly likely to being derived from a primary tumor source. And as you can see, tumor cells are very homogeneously distributed. Sometimes even similar cells are distributed here in defined patches. We can then also look into the other areas that are more likely to be from recurrent tumors, and we can already see that there is more noise, more different cell types, more stromal parts of the tumor, and also a couple of these purple cells here that containing more glial cells, astrocytic and oligodendrocytes. So as we can learn from this approach is already that there is a difference in the cellular compositions, rather the network predicts this as a primary or a recurrent source. So what we can do is now we can use in silico perturbations by having a primary microenvironment, and then we can use the classical techniques that we do in the lab. So we can spike in cells, we can deplete cells, for example, we can even exchange cells or rearrange the cells in space. That would be in silico perturbations that we can do on architectural level, but then we can also go to cellular level, overexpress genes, knock down genes, or even change whole chromosomal alterations within these samples. So these simulated samples then can be bring back to the network, and we can ask whether this change that we have induced by the perturbation would have any change of the network decision towards a primary or a recurrent stage. And this is one example in which we, for example, tried to understand how the transcriptional phenotypes are linked to epigenetic phenotypes. And there are 3 different epigenetic phenotypes in the GBM called RTK1, RTK2, and mesenchymal-like. And here we used a specific microenvironment, very typically from primary tumors, and we used astrocytic-like tumor cells and changed all these astrocytic-like tumor cells towards mesenchymal-like tumor cell identities. And here we can see that we cannot do this all in once, but rather we can do this in iterations. So within 52 iterations, we almost exclude all astrocytic-like origins from these tumor, and here already enhance a very mesenchymal-like tumor. And when we then use the network to predict whether this has any change on the epigenetic phenotype, we can see that after a certain amount of exchanges of cells from astrocytic to mesenchymal, we see that the epigenetic phenotype switches from RTK2 to an RTK1 tumor. So this already gives us an idea that we can use in silico perturbations to really understand how the tumor phenotypes might change. So we can use this already with all cells in the tumor microenvironment to check whether which cell has an impact on the decision of the network towards the primary or the recurrent stage of the tumor. And so all the cells that we can see having their peaks on the right side are being more likely to be enriched in recurrent tumors, and all that ones on the left side are being enriched in primary tumors. And there was not really a surprise that we can see that specifically CD8 T cells, macrophages, and mesenchymal-like tumor cells are enhanced in recurrent tumors. This is also described in the literature from single-cell transcriptomic data. But here we could identify that also cells like neurons and oligodendrocytes are very specifically enriched and more abundant in recurrent tumor microenvironments. So we looked there a little bit more in detail, and we observed that if we use a primary tumor microenvironment and we spike in oligodendrocytes, we can, over different locations of spiking in these cells or different primary microenvironments as a start point, we can see that we have very robust enhancement of the prediction score towards a recurrent tumors when we enhance oligodendrocytic cells within the tumor. So this already gives us a first idea that not only the change of the tumor cells are important, and not only the enhancement of CD8 T cells or T cells in general and macrophages are important to reshape a recurrent microenvironment. It is also neuronal cells that making a very strong impact here towards the recurrent stage of the tumor. We can also use this approach now, not specifically looking in tumor stages, but rather in different therapeutic approaches. And here we ask which microenvironment and cellular compositions of these microenvironments are likely to be enriched when we treat the patients with checkpoint inhibition. And we could identify that, for example, B cells is one of the classical cells that being enriched then when we do checkpoint inhibition, and when we even enriched B cells within tumor microenvironments, the prediction score towards checkpoint inhibition is growing and enhancing. We tested this now and validated this on single cell level and on the spatial transcriptomics, and we could see that if we use paired samples from primary and recurrent stage after PD-1 inhibition, that one of the cell types that really was enhanced and enriched in these microenvironments and in these tumors were B cells, plasma cells, and TC cells. So we could also see that the prediction that we made based on the AI model could be validated in real patient samples. So the second approach I want to talk about is the more passive approach, in which we use attention or integrated gradients or mathematical explanations of how a decision was made towards a primary or a recurrent tumor. And so one of the drawbacks of the using the Vizgen technology with a very low resolution is that we do not really know where are the cells localized on cellular level. We can see and look into microenvironments, but we don't know really where are these cells. So we cannot really specifically put importance of single cells more on specific microenvironments. So to overcome these problems, now we look into in situ sequencing data, in which we really get the single cell resolution, and we can really understand how the cells and the cellular interactions are participating in the decision of a network towards a primary and a recurrent. And similar to the Vizgen samples, also microenvironments derived from in situ sequencing data can be highly accurate, predicted, whether this comes from a primary or a recurrent status. And also, if we look then specifically on the latent space representation here as a UMAP, we could see that the stages of the tumor making a huge difference, but individual patients are not really participating in these decisions. So the network and the latent space is really reshaped to identify microenvironments from primary or from recurrent tumors, not really so much influenced by the patient or individual transcriptomic informations. So in this very complex overview of how these microenvironments can look into, we combine different informations that we now can obtain from the explainable AI approaches. One is the attention mechanism, so we can ask the network which cellular interaction, which cellular combinations are really important to make the decision whether this tumor microenvironment is derived from de novo or from recurrent tumors. And then specifically to put weights on which cells are important, we can use integrated gradients here so that we can see which of the cells are really being important when the network wants to make the decision towards a primary or a recurrent tumor. Now we can use this information and look them a little bit more specific into the importance of different cells. So these integrated gradients are basically giving us the information which cell of the network of the microenvironment is important for the network to make the decision towards primary or recurrent. And there is some cell types that are more important in general. So we see this here along this axis, that cells that being in the upper right square are being in general important to make a decision towards a primary or recurrent. And then when we come here to the cells being associated more to the bottom right or to the upper left side, these are cells that being more important for the specific stage of the tumor. And here, as we can see, that, for example, stromal cells, endothelial cells, and all vascular cells in general seem to be more important for de novo tumors. And then, as we have already seen before, cytotoxic T cells, but as well oligodendrocytes and neurons are being very important to make the decision towards a recurrent tumor. So, but the attention that we already obtained, as we can see a couple of slides before, now give us the information not only how important the cell is, but how important the embedding of the cell in this specific neighborhood is. And this gives us kind of an idea of which cellular interactions are being very important to make the decision towards a de novo or a primary tumor or recurrent. And what we identify here is that in primary settings, for example, it's more abundant and more connection towards myeloid cells, for example, connected as macrophages and activated macrophages and monocytes. And when we switch into the recurrent stage, or then we see that microglia and activated microglia here becoming more important to connect the tumor microenvironment. And also the part of the tumor is really changing. So we see an enhanced interaction and importance of oligodendrocytic-like tumor cells that are connected to microglia cells and oligodendrocytes in general. And we don't see this so much in a primary setting of these tumors. So there's a higher importance of these astrocytic-like and mesenchymal-like tumor cells being connected to each other. So by understanding now these tumor microenvironments, we really now need to take this knowledge and put it into clinical applications, how we can use this. And one of the things we developed most recently is something for a diagnostic purpose. So we can use now these spatial data to really trying for a neuropathology diagnosis, which can be then made on a single slide, for example, not using multiple next generation sequencing approaches and epigenetic profiles. So we can all use this from a single slide. And this is sometimes important, especially in brain tumors, in which we have only very few and very small tissue pieces, and we need to make a full diagnostic panel on these very tiny pieces of tissue. So here we really need to use the highest amount of data obtained from the smallest piece of tissue. And together with the neuropathology in Heidelberg, especially Felix Sam, we developed an approach called Napster, in which we can now use spatial transcriptomic data and basically get the whole panel on neuropathological diagnostics just within one slide of the tumor. And this is an example brain tumors are mostly identified and classified by the Heidelberg classifier, which is an epigenetic classifier coming from the Illumina epic array data. And we could see that if we train the spatial transcriptomic microenvironments to be identified and annotated towards these different phenotypes that we see on epigenetic level, we can see a nice overlap that also spatial microenvironments and spatial transcriptomics can be used to accurately predict which specific epigenetic phenotype of the tumor we have. And this becoming even more important if we want, for example, to predict specific genetic alterations within the tumor. And here, one problem that we have if we use spatial transcriptomic data or single cell data is that we can infer copy number alterations. For example, here, a very important loss in CDKN2AB, which is important to differentiate lower grade gliomas to be more malignant or non-malignant. We need to identify these deletions, which can be sometimes combined with a deletion on chromosome 9, but sometimes we have these as single gene deletions. And these single gene deletions are very difficult to detect. So we ask ourselves probably if there is a clinical difference of these patients and this character of this tumor is more malignant, there also might be a different microenvironment. And we could see that the microenvironment itself is very predictive for the specific heterozygote or homozygote deletions of CDKN2AB. And this is very important, especially when we want to specifically look into different biopsies of patients. So, for example, tumors in which we obtain now multiple tissue areas. And we want to see whether this specific deletion that makes the tumor more malignant is in, for example, some of these biopsies, we can see this deletion, and in other biopsies, we don't see this. And for a good grading or perfect grading of these tumors, we really would need to do next generation sequencing from all of these different biopsies. But by putting and multiplexing all these biopsies together, for example, we can see that we identify homo or heterozygote deletions within different parts of these biopsies that really helps now to make better decisions and better diagnostics for these brain tumors in general. So as a neurosurgeon, I'm also very interested in how we can now basically use these data to also make better decisions, not only for diagnostics, but make better decisions for our surgical approaches. And together with one of my colleagues in Hamburg, we have seen that potentially not all tumors, not all glioblastomas are really benefiting from a gross total resection. So the classical approach of these tumors, as we can see here on the left side, is we want to remove as a surgeon as much as possible of these tumors. So we call this gross total or maximal, supramaximal resection approaches. And we know this is at some point connected to better survival. But the problem is that the larger resection is, the higher the risk is for the patients to receive functional deficits or permanent neurological deficits. So it's always a question how to balance now perfect oncological outcome and the functional outcome of these patients. So there might be, for example, biomarkers or areas in the tumor that need to be resected more, and there's other parts of the tumors or specifically other tumors that do not really require a gross total resection. So to have now biomarkers to really guide surgical decisions, we wanted to see whether we can understand if there is molecular features that really can guide us to make a better decision whether some areas or some tumors require a larger or smaller extent of resection. So one of the things we identified was an epigenetic signature we call as the high neural signature, which really makes a difference whether this tumor is going to be fully resected with a gross total resection or do not, basically really benefiting from these gross total resection. As we can see here on the right side, on the survival course, there's a huge difference whether these high neurotumors are going to be partially or fully resected. But there's, on the other hand, there's other tumors that we contain as low neurotumors that do not really make any difference in the overall survival and outcome, whether there is a resection, a fully resection, or almost a partial resection of these tumors. So we again wanted to use now spatial transcriptomic data to understand what is now the difference between these more neuronal-like tumors and the non-neuronal tumors. And we can again use our network approaches to specifically predict now microenvironmental features and compositions that really make the difference between these neural high and neural low tumors. And we could identify that there is a higher abundance and connectivity of these high neural tumors towards the neural system. So there is a higher amount of synaptic contacts and input of the neural system towards the tumors. And so these tumors are benefiting more from a gross total resection. And on the other hand, these low neurotumors having a higher abundance of immune cells. So there's a higher immune reaction in general of these tumors. And we could see that these tumors that having a higher immune response basically not benefiting from gross total resection compared to these high neurotumors. So we can in general use now spatial transcriptomic data to identify biomarkers and especially understand biomarkers that we can then use to guide clinical decision, make better decision, not only on the therapeutic or diagnostic approach, but also for surgical guidance and surgical decisions. We really can use molecular data to optimize our performance as clinicians. So with this, I want to close and really want to thank the organizers of this webinar to share all these results, and really want to pinpoint that we need further validation and integration of molecular features to understand these brain tumors, and then creating AI approaches, advanced machine learning approaches. Now how we can use and transfer this molecular understanding, this architectural understanding of these tumors to further make better decisions for the patients, to personalize the therapy, and also to really enhance performance of surgical guidance and decisions here. So with this, I want to thank, and this is my team, my different labs in Freiburg and in Erlangen, and also all collaborators that really participating a lot in all this work that I presented. So with this, I want to thank, and I'm really happy to take some questions. Thank you.
All right. Well, thank you, Carole and Henrik, for your informative presentations. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. So our first question asks, could you detect splice variants?
Hello. Yes, it depends on the sequence of your different isoforms, and we do a custom design. We've also got a new chemistry coming in that should definitely improve this, so we can absolutely work on that.
Great, thank you, Carole. All right, next question here. How big is the MERScope imageable area?
It's a great question. As of today, as of recently, it was 1 square centimeter, but we've just announced our new instrument, MERScope Ultra, that now is going to be able to accommodate up to 3 square centimeters.
Great, thank you. All right. Another question here. How long is the runtime for MERScope?
Yes, so for Ultra, at the highest plex that we have, with all the cell boundary, all the proteins that you could add, and the maximum square, like 3 square centimeters, it would be 48 hours. And if you're on normal MERScope and you're still at the highest plex for everything, it would be 24 hours.
thank you. Now, what is the maximum plex available?
We have a fully custom 960 gene panel available for people, so you can fully design it and be able to detect all of these genes.
Great, thank you. All right, now how do ex vivo analysis of tumor cell types transcriptomic could drive in vivo surgery?
It's a very interesting question. So there's basically no direct answer, but what we believe or what our analysis was basically done on surgical specimens. So we guess that the definition of the cell types that we see on specimens is very likely to be the same that we have in the surgery. So the idea is basically to generate biomarkers to understand these architecture, to really know which of the tumors benefiting more from gross total or benefiting more from a larger extent of resection in within specific areas of the tumor. So the idea of having one concept fits all, so every area needs to be resected similarly, is potentially not that what we would like to look into, but more specifically to understand which area might be more important to be resected than other areas. And one of the very important question here is how we can transfer this information completely into the OR. I think this is something we're working on intensively, but this is the general idea and concept.
Great, thank you, Henrik. All right. Another question here. If we take into consideration that oligodendrocytes or neurons are involved in the recurrence of tumors, could specific genomic identity from neurons located at the vicinity of the tumor compared to neurons from healthy parts of the brain be obtained by spatial transcriptomic?
Yeah, I think there's definitely the limitation right now that we have in the technology to look into genomic features here. So I think this is nothing we can really look into. We just use these technology that we have right now. If you think about a 500 or 1,000 plex panel, to specifically identify subtypes of cells. So we cannot really do data-driven analysis like we do in single cell or in Visium data with these specific informations. But we notice that basically the information that, for example, the neural network is using to guide the decision is basically the cell type identification. So we have basically trained on transcriptional information or trained the network on the information of the cell types alone, and the performance very similar. So I think here it is more how the microenvironment is defined on cell type identification and subgroups, rather than more on the pure transcriptomic information. And I think this is a little bit the difference between the technologies that we have. And this is also the importance to use multiple spatial technologies, because we can really combine the depth, for example, that we see in Visium data with the detailed information in terms of resolution that we get from Merfish data. So I think that's the beauty of integrating all these different methods.
Great, thank you. All right. Now we have a question regarding the sender and receiver cells. Can you explain how those are defined? Excuse me. Is it by their clustering next to each other in a spatial context?
Yeah, that's a very important question, and I think I heard this a lot. Sender and receiver potentially might not be the correct wording, because at the end of the day, we have not a very defined way of doing a direct graph representation. So the idea right now is having a directed graph from the center towards the neighborhoods. So whenever there is a connection from center towards the outside of the center, we have these kind of a direction here, which is actually not a functional connection. It's more a spatial localization or a spatial direction. And then how we define these different receiver and sender cells is then we quantify across different microenvironments the amount of attention that was put towards two different cells. And whenever a signal derived from inside to outside, so there is basically the directed vector how we define these cells. So it's not a functional way, it's more a spatial definition of sender and receiver. It's basically probably not a good word or not a good explanation for that, but I think we need to work on a better wording there, definitely.
Thank you, Henrik. All right, we've got some great questions still coming in here from our audience and some more time. So we have a few more minutes to answer a few more. So we'll continue. This next question asks, are the prices for Merscope Ultra available on the website for different panel sizes?
Maybe not on a website, but feel free to just reach out to us and we'll certainly be able to help you.
Great, thank you. All right. Now, in a spatial context, the MES cell state of glioblastoma seems to be connected to necrosis. Would you say that the methylation class and switched due to proximity of necrosis?
I think yes, very, very important to make clear that transcriptomic state of mesenchymal-like cells is completely something different than the epigenetic status. Yeah, so the epigenetic status of a mesenchymal-like tumor is basically linked to its immune compartment. I think that there's a couple of works that are ongoing, and I think also some that are published already, that it's very clear that the epigenetic mesenchymal-like phenotype is something that represents the immune compartment of the tumor. So then we come back to two RTK phenotypes, as I mentioned, and I think here we have a little bit the difference between connecting these to the mesenchymal-like phenotype and the neuronal more like different kinds of OPC, MPC-like cell-like phenotypes. At the end, the question of necrosis is something that we see there's a connection between mesenchymal-like cells and necrosis. This is completely right, and this is what I described in the very first slide, that there are different parts of reactive programs. There's two different ones, that mesenchymal-like cells can be linked to immune response, but also can be linked to metabolomic alterations such as hypoxia, and so that's a very specific part of the mesenchymal-like cells are definitely linked to necrotic areas or near necrotic areas, but there are also other mesenchymal-like cells that are linked to immune response areas. So it's not that mesenchymal-like cells in general are only localized next to necrotic areas.
Great, thank you. All right, next question here. As some GBM are radio resistant, have you looked if you could have some effect of the tumor environment in this resistance? And if yes, are there any type of cells that have a more important role in this resistance or in the reoccurrence linked to this resistance?
Yeah, definitely. So this is one of the major purposes of this project, to not only identify what is important for primary and recurrent tumors, but also what is important for different time points of recurrence, different resistance mechanisms, and so on and so on. So we're looking into this. I cannot really give right now a completely explanation of how this appears. And also the problem is that in patient data is very difficult to look specifically in the ones that have raised, like, because we always get chemotherapy and radiotherapy together. So it's basically very difficult to say what is the part of the radiotherapy, what is the part of the chemotherapy, because this two protocol combines both together. But at the end, this is something we're going to specifically try to understand how the resistance mechanisms are appears, and if this is always a similar mechanism, or are there different kind of meta mechanisms that we can identify that different patient groups, for example, receive similar pathway signalings in terms of resistance. But yes, this is part of the project, actually.
Great, thank you. All right, now can this prediction method be used to compare different data sets before and after irradiation of cell lines, and what validation data should we use?
Yeah. So the beauty of this system is to being used directly from tissue specimens. Yeah, I think from cell lines, it's always a little bit difficult, or it's not really makes sense to use spatial methods here if you look specifically only on cell lines. So that would be more interesting to be compared on single cell data level or something else. So here, these methods are more specifically designed to look into real tumors, to understand the complexity of microenvironments and their change. And yes, there is the possibility to use the same network approach, the same AI approach to compare, for example, various of different cancers, and then specifically mention, for example, different types of the cancer as confounders. So because this is kind of the idea that the discriminator can also remove all these confounder information that we do not want to look into, so specifically want to identify what happens, for example, after radiation or something like this. So that is possible, but highly recommended to being used more for complex tissues rather than for cell lines alone.
Great. Well, thank you, Henrik. And that will actually conclude our session for today. So Carole and Henrik, do you have any final comments for our audience?
Yeah, thank you very
No,
much. It was
yeah.
really a nice.
Sorry.
I'm sorry. Really nice webinar. And now it's like, yeah, I'm sorry. Your comments.
No, I was going to say the same. I was going to say thank you, everyone, for connecting for all your very interesting questions.
Great. Well, thank you again, Carole and Henrik, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Vizgen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.